Hello students, welcome back to our SEMT YouTube channel. Today we are going to discuss about the revision of chapter number 5, that is principles of inheritance and variation. Now you are already knowing that this chapter is based on the heredity and variation. So you are familiar with the term heredity. When the characters are transferred from parents to offspring, that is called as heredity, and the process of the transmission is referred as the inheritance. Next thing is about the variation. What does the variation means? As I said in our live lectures, variation is the degree. It is the degree of the difference between the progeny and its parents. That is called as variation. So this was the first two basic terms of the chapter. You are familiar who is the father of the genetics. Father of genetics is Gregor Johann Mendel, and you are also familiar the father of modern genetics is William Bateson, that we have already discussed in our Zoom live lectures. Now talking about the first experiment of the Mendel selected pea plant for his experiment, right? And the pea plant scientific name is Pisum sativum or Pisum sativum, right? He selected this plant because this plant was having a shorter lifespan. Okay, means you can get the generations very early. Shorter lifespan, it was annual plant having 2 to 3 months lifespan in which it produces the offsprings. Second, self pollination and self fertilization was very easy. It was natural process in pea plant. Artificial hybridization was also easily carried out. After that, this was easily cultivated, and very important, pea plant was having many pairs of contrasting traits, and Mendel selected 7 pairs of contrasting traits for his experiments. So this was about the selection of pea plant. Now Mendel conducted his experiments from the year, which is very important. It was a gap or period between 7 years. His experimental years was about 7 years, from 1856 to 1863. This was the period of 7 years in which he conducted his famous hybridization experiments. First experiment was monohybrid experiment. Monohybrid experiment. Right? In monohybrid experiment, he selected or he considered only single character inheritance. That is, study of inheritance of a single character. Right? So students, this we have already discussed. This is just a revision video. Right? We are just revising the topics which has been taught. He selected single character and he studied the inheritance. So first of all, he took a parent generation, that is capital T, a tall plant and a dwarf plant. Now you are familiar, this will produce one type of gamete, right? This will produce only one type of gamete, as 2 raised to n becomes 2 raised to 0, that is 1. So when you are getting F1 progeny, all plants will be having one capital T and one small t, that is our F1. So in case of F1 plant, we will get all the plants tall, right? He got all the F1 plants tall. When he self-crossed, right, he got F2. In F2, see, in F1 generation, the dwarf character of the parent was not seen. But when he got F2, right, this is Punnett square or checkerboard method, which was given by Reginald C. Punnett, right? These are the gametes. He got ratio like this, both capital T tall, capital T small t tall, and another one, last one was dwarf. So he got to know that the parent character which was, or trait which was dwarf, was not seen in F1, but that trait was again recovered in F2, and he got ratio of 3:1. That means he conducted the experiment and he selected 1064 plants out of 1064 in F2, right? Number of plant when in the ratio of 3:1, that is approximately 787 plants, right? And 277 plants. 787 were the tall plants, while 277 were dwarf, which was near about the ratio, that is 3:1. And this was phenotypic ratio. This was phenotypic ratio, means when we see from the external side, the external features, 3 plants were tall compared to the dwarf, there was only 1. Now when we talk about the genetic constitution, genotypic ratio was different, that is 1:2:1. How? So capital T, capital T, capital T, small t, and small t, small t. This was the ratio observed in the monohybrid genotypic ratio. After this first experiment, he proposed law of dominance and law of segregation. So these two laws were proposed by Mendel, right? This we have already discussed in very detail, all the 3 laws, right? The first proposal was like that, first conclusion, that characters are controlled by discrete units called as factors. So the term factor was said by Mendel. After that, second law, characters occurs in factors occurs in pairs. So that was the second one. Third one, second conclusion was that when the two unidentical or non-identical factors of a unit pair are present in the hybrid, one is dominant, another one is recessive. So this law was based on F1. When two unidentical factors of a unit pair are present in a hybrid, so these two are unidentical factors, they are present in a hybrid, capital T is dominant and hence it expresses itself, and that is tall. Small t is recessive, so it gets suppressed, right? So in this case all plants were seen as tall, right? Now next one was law of segregation. During the formation, see in this case law of segregation says that alleles do not blend with one another. Alleles do not blend with one another, and during the time of formation of gametes, each factor of a unit pair enters into a separate gamete, right? So means they segregate at the time of gamete formation. This was the law of segregation. So this is about the monohybrid experiment, right? After that we will see about the test cross, right? So what is test cross? first we need to understand the first thing, that is the back cross. Okay, so let me clean this and then we will understand the back cross and its two types. Okay, first one is back cross. When the F1 individual, right, F1 individual is crossed with the parents, that is called as back cross. When F1 individual is crossed with parent, right, any parent, it is called as back cross. Now it's are two types. First one is out cross. In out cross, I have already taught you what happens in out cross. This is not in the textbook, but I have explained it. Then F1 individual is crossed with the dominant parent. You will get all the plants or all the types will be dominant because the parent is homozygous dominant. In case other type is test cross. Test cross is opposite to this. F1 individual is crossed with recessive parent. Yes or no? Yes. See, this is dominant. In this case, it is recessive. Test cross helps to determine the dominant genotype of the individual. Remember this. Helps to determine dominant genotype of individual. This we have done experiment. Let's see again. If a plant is having a dominant character, let's say that the plant is having dominant trait, that is plant is tall, right? So if I say plant, let's say plant is tall, but for tall there are possible two types of genotypes. Yes or no? Yes. That is capital T or capital T small t. There can be two possible genotypes. Now, out of these two genotypes, which genotype is present in the plant, that we need to discuss. Yes or no? We need to know that which is the possible genotype. So in this case, the plant is crossed with the recessive parent, that is small t. So it is a type of test cross. This is the F1 individual, right, having capital T or small t, capital T small t. We are not knowing this. Yes or no? So when we make a cross, we can get two possibilities. First, if the plant is having capital T, then we need to cross this. So the possible thing will be like this. Then capital T, small t. So we get all the plants tall. If all plants, when you cross the plant with the recessive parent, if all plants are tall, then the genotype is this. If you are getting F2 like this 50-50, then the plants are not homozygous tall. Plants will be heterozygous tall. Let's say small t, capital T, small t. So over here you can get these two tall and these two dwarf. Yes or no? So it is 50% tall and 50% dwarf. Then the plant genotype will be this one. So are you clear why the test cross is performed? Now we will discuss about the next topic, that is incomplete dominance, right? Let's say now when the Mendel proposed law of dominance, right? This is actually after the work of the Mendel was rediscovered by three people, Carl Currens, Hugo de Vries and von Scherman. These people rediscovered the work in 1900 after the death of Mendel. Now, the law of dominance was proved wrong in certain cases. It was having exceptions. So, that exception is referred as incomplete dominance, right? In case of incomplete dominance, the dominant factor is not completely dominant over the recessive one, and hence the character is blended, right? There are many examples. Let's say Mirabilis jalaba or Enterinum majus, right? Enterinum majus. This we are showing. Even the textbook example is that starch grain size, right? We can take starch grain size. Now, let's say this, right? There were two types of plants. Some were bearing red flower and some were bearing white flower. That is, RR is referred as red and small r, let's say white. This is a parent generation. Now, first progeny, that is R small r. Now, according to Mendel, the plant having capital R means it should bear which type of flower? Red flower. But in these plants, there was seen a blending of character. I am saying the word alleles are not blended. Remember this. This is just a blending of characters. So the plant was F1 progeny was not having any of the feature like the parent generation. So when again that self cross was made, right? In this case, both the parents' characters were recovered in F2. Capital R, capital R, smaller. So in this case, both the parent characters were recovered, right? And the ratio which was obtained was both same. See, let's say phenotypic ratio. Now, in case of phenotypic ratio, we can say we got one plant red, two plants pink, and again one plant white. If we talk about the genotypic ratio, it was also same. Reason was 1:2:1 because one plant was having capital R, another one capital R smaller, and another one was smaller. So this was the exception of the law of dominance. Same way, we also got another exception in which both the alleles becomes dominant, and so the F1 progeny is showing the characters of the both one, that is referred as codominance, right? As I said, in case of codominance, we can take AB blood group. Yes or no? I have explained it. This entire ABO blood grouping, multiple alleles and codominance together, with taking entire lecture on it. So I will not go that much deep now. I am just discussing about the ABO blood group. AB blood group is, its genotype is IAIB. As both the genes are dominant on the surface of RBC, we are able to see both the type of sugar polysaccharide, that is antigens, and there are no antibodies. When these both are dominant, we are able to see both the antigens. This is also another example of the exceptions for law of dominance, because both the genes become dominant and we are able to see both the antigens. So this was about the today's revision topic. Thank you.